This will be lecture 7, and we were studying carbohydrate metabolism. Okay, so in the last class we studied glycolysis, and today we will study some other parts of the glycolysis. Okay, so what are the importance of phosphorylated intermediates? So you might see 9 out of 10 glycolytic intermediates between glucose and pyruvate are phosphorylated. The phosphoryl groups seems to have 3 functions. The first one, because of the plasma membrane generally lacks transporter for phosphorylated sugars, the phosphorylated glycolytic intermediates cannot leave the cell. After the initial phosphorylation, no further energy is required to retain phosphorylated intermediates in the cell. Phosphoryl groups are essential component in the enzymatic conversion of metabolic energy. The energy released in the breakage of phosphoanhydride bonds, such as in ATP, is partially conserved in the formation of phosphate esters, such as glucose 6-phosphate, and high-energy phosphate compounds formed in glycolysis donate phosphoryl group to ADP to form ATP. And third is binding energy. Binding energy resulting from the binding of phosphate group to the active site of the enzyme lowers the activation energy and increase the specificity of the enzymatic reactions. The phosphate groups of ADP, ATP, and the glycolytic intermediates forms complex with magnesium, and the substrate binding site of many glycolytic enzymes are specific for this magnesium complex. Most glycolytic enzymes require magnesium for activity. So those are very important three intermediates, three importance of why phosphorylated compounds occur in glycolysis. So the first one is that I summarize is very important to retain. Phosphorylation is very important to retain the glycolytic intermediates in the cell. And secondly is provide phosphate group to produce energy, and thirdly it increased the enzymatic specificity. Okay, so what are the 3 catabolic pairs of pyruvate? So there are 3 pairs. The first one is in case of oxygen or in aerobic respiration, the glucose is converted to pyruvate in cytoplasm, and then pyruvate will enter into the mitochondrial matrix, and there it will be converted to acetyl-CoA, and then acetyl-CoA will enter into the citric acid cycle to produce carbon dioxide and water. And in case of anaerobic respiration in mammals, pyruvate will be converted to lactate in vigorously contracting muscle or in erythrocytes or in some other cells, and also in some microorganism. So this is the aerobic fate and this is the anaerobic fate. Thirdly, there is another anaerobic fate of pyruvate in lower microorganism like yeast. It will be called fermentation, where pyruvate will be converted to ethanol and carbon dioxide, and this is the third phase. Okay, so the oxidation of pyruvate is very important catabolic process, but pyruvate has anabolic fate as well. It can, for example, provide the carbon skeleton for the synthesis of amino acid like alanine. So fates of pyruvate under aerobic and anaerobic conditions, I think I have already discussed those 2 important things in the lecture 6, where I said the utilization of NADH or the oxidation of NADH to NAD is very important to continue the glycolytic reactions in aerobic conditions. But in anaerobic respiration, the NAD plus will not be formed, so the NAD plus have to be formed in several other reactions to continue the glycolytic pathway. So let's study those. So pyruvate is a terminal electron acceptor of lactic acid fermentation. So when animal tissues cannot be supplied with sufficient oxygen to support aerobic oxidation of the pyruvate and NADH produced in glycolysis, NAD plus is regenerated from NADH by the reduction of pyruvate to lactate, where pyruvate is converted to lactate through the lactate by the lactate dehydrogenase enzyme, where NADH is converted to NAD. And another case, so yeast and other microorganisms ferment glucose to ethanol and carbon dioxide in 2 steps. In first step, pyruvate is converted to acetaldehyde by decarboxylation reaction, and this is a reversible reaction catalyzed by pyruvate decarboxylase. And where in the second step the acetaldehyde is converted to ethanol by alcohol dehydrogenase, and where NADH is oxidized to NAD+. And in this reaction, the first step, there is a very important molecule, TPP. This is derived from vitamin B1. This is a coenzyme, and also magnesium is used as a cofactor here. So thiamine pyrophosphate is very important, and in human, if the diet lacks of vitamin B1, it can produce very which is characterized by the accumulation of fluids or swelling and pain and paralysis, and ultimately death in the human being. Ultimate death of a human being. Okay. So some reactions in which TPP is an essential cofactor. Here are the enzymes and here are the pathway. You might be interested to memorize those ones, like pyruvate decarboxylase enzyme used in alcohol fermentation, pyruvate dehydrogenase enzyme used in succinate, used in synthesis of acetyl-CoA, alpha-ketoglutarate dehydrogenase used in citric acid cycle. Those all the enzymes are using TPP as their essential cofactor. So what are the feeder pathways of glycolysis? That means we studied glycolysis in the last lecture 6, where we studied just glycolysis has been initiated from glucose, but there were other carbohydrate or other members of carbohydrate. So in this slide, we will study how the other carbohydrate will be entering into the glycolytic pathway. So let's start with the glycogen. Glycogen or starch will be converted to glucose 1-phosphate, and glucose 1-phosphate then converted to glucose 6-phosphate. Thus, it enter into the glycolytic pathway. And for mannose, they can first produce mannose 6-phosphate, then it is converted to fructose 6-phosphate. Phosphomannose isomerase enzyme will be responsible for here, and thus mannose can enter into the glycolytic pathway. So lactose, it contains glucose and galactose. So when galactose derived from lactose, it will be converted to UDP galactose and exchange with UDP glucose. Then UDP glucose will be converted to glucose 1-phosphate. Then glucose 1-phosphate will be converting to glucose 6-phosphate. Then it will enter into the glycolytic pathway. And glucose, directly can be entering into the glycolytic pathway using glucose 6-phosphate. So what about fructose? Fructose might be entering into the glycolytic pathway in two ways. One is fructose to fructose 6-phosphate, then fructose 1,6-phosphate, then enters into the glycolytic pathway. Or in other case, fructose can be converted to fructose 1-phosphate, then it is converting to glyceraldehyde 3-phosphate and dihydroxyacetylene phosphate, and those two will be converted to glyceraldehyde 3-phosphate. So let's study in details. So D-fructose is present in many fruits in free form and also by the hydrolysis of sucrose in the small intestine, and it can be phosphorylated by hexokinase. So in muscle and kidneys of vertebrates, this is a major pathway where fructose is phosphorylated to fructose 6-phosphate, then enters into the glycolysis, and the reaction is catalyzed by hexokinase with the presence of magnesium. In the liver, fructose gain into the glycolysis by phosphorylation of fructose by another enzyme, fructokinase, at carbon 1 position, and then fructose 1-phosphate will be converted to dihydroxyacetylene phosphate and glyceraldehyde, and then glyceraldehyde will be converted to glyceraldehyde 3-phosphate by kinase isomerase enzyme. Thus both products of fructose hydrolysis enter in the glycolytic pathway as glyceraldehyde 3-phosphate. So what about the galactose? So here galactose will be converted to galactose 1-phosphate, and galactose 1-phosphate will be exchanged to glucose 1-phosphate, and then it will convert it to glucose 6-phosphate, then it will enter into the glycolytic pathway. So where UDP glucose is used here to transfer the glucose, then converted to galactose. This galactose can be converted to glucose by UDP galactose epimerase enzyme. So it is a very important monosaccharide and important in human nutrition, and it is converted to glucose 1-phosphate in the liver and subsequently stored as glycogen. So other case, the mannose. Mannose is arise from digestion of various polysaccharide and glycoproteins present in food, and this can be phosphorylated at carbon 6 position, producing mannose 6-phosphate. Mannose 6-phosphate and then is isomerized to phosphomannose mannose 6-phosphate, then converted to fructose 6-phosphate by phosphomannose isomerase, and fructose 6-phosphate then converted to fructose 1,6-bisphosphate, and then enter into the glycolytic pathway. This is the feeder pathway of glycolysis. Through all other member of carbohydrate can enter into the glycolytic pathway. So that's all for this class.